Hey there, welcome back to our second part of the video on kidneys and nephrons. And today we're talking about the process. And so we are going to, in the last video we just labeled everything. And so now we have an understanding of how to use the words, and now we can talk about what's actually happening. So if you remember, this is a nephron. So this is let's just call this a tiny nephron. It is just a very basic format as to what's happening inside the nephron. And so here we have our capillary. So remember, this is our afferent capillary. So the blood is going into the capillary, into the glomerulus, and it's coming out. And this is the efferent. And so remember that your blood is pumped by your heart, and the pressure of your heart pumping your blood is going to push it through and into this glomerulus. And the glomerulus is going to be very porous right here. And so that blood, about 1/5 of that blood, is going to be pushed through and to become filtrate. And so let's just put 1/5 here. And so filtrate is going to be the substance that's passing through your nephrons. And so here, as it's passing through the nephrons, it's the good things that we have that we filtered through. So this is not a very selective filter. It's going to push good things and bad things through, or stuff that we want to keep and stuff that we want to get rid of, which is a better word to say than good or bad. So it's going to push things through like glucose. It's going to put vitamins. It's going to push vitamins through. It's going to push through salt, and it's going to get rid of stuff that we don't want, like ammonia. We don't want ammonia, or and other materials. And so those things are the good things that we want to keep are going to be reabsorbed. So they're going to be reabsorbed back into the blood. And so in between here, we have the transport epithelium. So here, this is going to be in between the tube in which the filtrate is being passed. We're going to have a transport epithelium, or just here we go, let's just call this the tissue of what is the kidney itself. And so materials are going to pass they're going to be reabsorbed through the transport epithelium and then into the bloodstream. And so sometimes the solutes are going to pass into tissue and stay there, and then water is going to pass through the tissue and go back into the blood vessels. So here, reabsorption of good stuff is going to happen. So that stuff is going to be reabsorbed through the transport epithelium and then back into the capillaries. And so we talked before about how we had the vasa recta and we had the proximal and distal tubules. And so those are the capillaries that we're going to be talking about. So we have reabsorption, but then as we go through, we're going to see that there's going to be more ammonia, more things that we want out. Sometimes maybe some sodiums are going to be passed out. So we're going to secrete. So from the transport epithelium, we're going to be secreting the toxins that we want to get rid of. And so as it goes through, it becomes more and more concentrated, more and more of toxins are being removed, and all the good stuff's coming out. And as it leaves the nephron through the collecting duct, so over here, the collecting duct, which then leads to the renal pelvis, which then leads to the ureter. I don't know why it does that. Ureter, and then urinary bladder. So that's the big picture. Now let's go a little bit past through this. Let's go past. Actually, let's talk really quickly. Let's remind you that these nephrons that we're talking about, here's the glomerulus, and it's going to pass down into the medulla, come back into the cortex. So that would be a cortical nephron, and remember that our juxtamedullary is going to be here. So I don't know where that went, but it disappeared. We're going to go down here, and so here it is. Here is our afferent. Here's our glomerulus. Here's our Bowman's capsule. And so our filtrate is passing through. And so here we have our proximal tubules. And we know that we have our peritubular capillaries around here. And then we have our vasa recta all around the loop of Henle. And so blood blood is going to be filtered through, and remember 1/5 passes through. And so big stuff is going to pass through, is going to stay in the glomerulus, is going to stay in the blood circulation. So large things like red blood cells, white blood cells, proteins, those are all going to stay in the blood. Individual amino acids are going to pass through. So things that are actually going to pass through, small things like glucose, amino acids, salts, ammonia. And so here we're going to have reabsorption. So reabsorption is going to happen in a pretty big way here. We're going to have active processes. We're going to have pumps, active transport using pumps to pump glucose back out to save the amino acids. Those are going to be pumped out. And so it's going to be pumped into through the transport epithelium, and it's going to then be put into pumped into the proximal tubules. And so here, the peritubular capillaries. And so we know that the filtrate's going to pass down here, and it's going to go into the descending loop of Henle, or the descending tubule. And we said here that there are aquaporins. And notice here that we have, that I drew an increase of solutes at the bottom here. And so we're going to have an increase. So the amount of solutes increases as it drops into the medulla. So here, we will say that it has about the concentration of about 400 milliosms, M-O-M-S, osms. And so down here, 600 milliosms, 800, and down at the bottom, milliosms. Down here, it'll be about 1200 milliosms. And so here we can see that there's a higher level of concentration of salts on the outside, and we have aquaporins, which is going to allow for water to be transported out. This is really important because so much if we excreted or peed out all of the water that traveled into a nephron that became filtrate, we would lose so much of our water. And so the nephrons are a huge player in helping us to keep our water. So here, the fact that we have aquaporins on this side is going to allow the water to pass back out. We get to save the good stuff, and that means we're saving the water. We are saving the water, and water is going to be pumped back into the vasa recta. So it's going to pass through the transport epithelium and then into the vasa recta. So we're saving the good stuff. And then over here we have the ascending tubule, which we said before is impermeable to water. But what we do have are pumps. So we're going to actively pump, meaning we're going to use energy in ATP. We're going to actively transport salts back out. So our bodies our cells can more easily pump solutes, the sodiums, the chlorines, etc., than it can water. So water has just always passed through our cells pretty readily, so we don't have pumps for water. So we have to create an environment that is highly concentrated. And so we have pumps over here where we're going to take the salt out, and we're going to take solutes out of the filtrate, and we're going to make this environment out here. We're going to actively pump salts out here to increase the osmolarity, or to increase the concentration of the medulla, so that it will continue to pull water out of the descending limb. So here on this side, we're going to be pulling salt out, and here on the other side, we're going to be pulling water out. So this is going to be passive transport, and this is going to be active transport. And so as our filtrate travels back up, and it's going to reach the distal, I don't know why that was so hard, the distal tubule, pretty much the same thing is going to happen here. We're going to have, once again, we're just going to try and take out all of the good stuff. So we're going to try and reabsorb the glucose, amino acids, anything that we don't really want to get rid of. We're going to try and reabsorb those. However, we're also going to be sending toxins out. So all the while we are sending toxins back out. So we are secreting toxins out, and we're trying to send this filtrate out into the collecting duct. And so once it's in the collecting duct, you would think that our job here is done. However, there are aquaporins here, lots of aquaporins here. I don't know why my pen has stopped working. Here we go. And this is going to be to allow for water to be absorbed at different rates. So our hormones, and we'll talk about this in 44.5, depending on what hormones are passing through our systems, more of these are going to be open, or more of these are going to be closed, and that's going to allow more water to pass through when we're not dehydrated. And if we are dehydrated, then less water is going to be able to pass through. And so that is going to then lead, so this is our collecting duct, and that's going to lead to the renal pelvis, where all of the collecting ducts end, and then that's it, and that is our process. So what I want you to do is to draw this out and come up with questions, and I will see you guys in class.